am Sonja Grün. I studied actually physics. And I moved at the end of my diploma thesis, which it was called at that time, to computational neuroscience, which was at the beginning of the 90s. And this field was just born, so to say. And since then, I also did a PhD in physics, but already with work in theoretical neuroscience. And yeah, I moved a bit more and more towards understanding. Sometimes I call myself a theoretical experimentalist because I did experiments on monkeys myself during my postdoc in order to understand what I'm dealing with. Yeah. So thereby I gained quite a bit of understanding what the pitfalls of experiments and so on are. And what is very dear to my heart, I will tell you about today. Yeah. And this is about analyzing massively parallel spike trains for signatures of functional ensembles. Now, our natural behavior is typically dynamic and fast. So you, for example, if you make a game with a ball, you have to catch the ball. Everything is the action is being done in milliseconds, hundreds of milliseconds. Or if you are looking, as you are doing all the time, looking around with saccades and fixations, even if you don't notice, such a sequence of a saccade and a fixation is about 250 milliseconds, which is fast. If you think about that, when you fixate with your eyes on something, you also have to decide where you go next and digest what you saw. Right? So there is not so much time. There were also there was also work by Simon Thorpe where he made experiments with humans and monkeys where he asked whether in a shown image, which was shown for 20 milliseconds, to identify whether in this image was a real animal or, for example, a picture of a deer on the wall. And people and monkeys were able to do this in very quick time, 280 milliseconds in humans, 200 in monkeys. So he made a calculation at the time, how much time you actually have from visual input to digest this and to perform a movement, and assuming about 20 milliseconds for each cortical stage, or you could also have less but more areas. But this with a typical firing rate of neurons, this leaves you about 1 spike per area. This is not much, right? And the question is how this should work. And I follow the hypothesis that neurons coordinate their activities on short timescales in a dynamic fashion. If you look at the cortex, which I'm mostly talking about, there are many neurons and many connections, which is also quite interesting that you have about a neuronal density of 40,000 per cubic millimeter. It's very dense. You have per neuron on average in humans between 10 and 20,000 silent. You know what it's like? Yeah, okay. So contact from other neurons to an individual neuron. This is also quite a lot, right? And an individual neuron is projecting to about the same number of different neurons. So what you have, at least in cortex, you have a heavy mixing machine, yeah, where you have a huge convergence on each individual neuron and a high divergence from each individual neuron. How can this how can that be made sense of? Yeah. And this speaks very much to my understanding for network processing. Now, there was a hypothesis, I like the figure very much, but this is an older hypothesis than by Ken Harris, that cell assemblies, groups of neurons, act as building blocks for information processing. This was brought up by Hebb already in 1949 in his book. I forgot. A very famous book. He was a psychologist, and he was thinking about how you could, so to say, process information. And he proposed already that processing by neurons is done by interaction of neurons. And taking this a little bit forward, so cross-neuron correlations are indicators of assembly membership. And I will push this further and further. So let's assume we have these subnetworks, as we can see here, where the first gets a sensory in. Can you see my cursor? Yeah. Gets sensory input to the first group of neurons. This is then propagated through a next subgroup of neurons and to a next. And I personally would call this whole, so to say, set of neurons involved in this current information an assembly. Now let's think about what we could measure. I always take the perspective of measuring on individual neurons level. Yeah. So we talk about neurons, yeah, and now we have electrodes. You probably know that you can do extracellular recordings where you can measure the individual spiking activities of the neurons. And what you could do or imagine is that, for example, you have only your electrodes here within this group of neurons, and you capture 4 of them which are involved in this propagation, and then you would notice this here in, for example, as a function of time. Here you have different neurons. Each line is an individual neuron. Each dot is a spike of that neuron. This is a sketch. That then these neurons, which you recorded from, are more or less synchronously active whenever you activate this cell assembly. I'm talking here on a time scale of 5 milliseconds or so, synchronous within 5 milliseconds. Now, you could also imagine a different expression of this cell assembly activity if you had the chance to record not only from this neuron, but also from this group of neuron and from this group of neuron and yet a further one. Then you could notice that neurons of this group are synchronously active, then with some delay, a group of neurons within this group, and so on. This is a hypothesis, yeah, which I'm working with. Now, however, there is a small issue. When we do electrophysiology, even if we record with 100 electrodes, let's say a Utah array or others, let's assume this would be such a Utah array. Each of these little dots are an electrode. They have a distance of 400 micrometers, such that you cannot record. You can record a neuron from one, which is close to one electrode, also on the other. There are other electrodes, as you might know. However, let's assume this, yeah. So you have 10 by 10 electrodes. These kind of Utah arrays exist. And then let's assume this is your, so to say, your assembly groups of neurons where the activity is propagating through. However, you can only record from a small fraction of these neurons. For example, the ones which are pretty close to one of the electrodes. So your result is you have these groups of neurons, but at the very end you record from 2. So this issue of subsampling is something which we always have to deal with. And just to give you some numbers, with this 100 electrodes, you sample about roughly 100 neurons from about 30,000 in this area. Yeah, so if you hear this, you could think you will never see anything about this processing. Okay, let's go back. So I said this might be the propagation of activity, and our subsampling likely does something like this.
Is the recording from individual neurons or superposition of a group of neurons?
I skipped all these spike sorting issues, yeah, because there I had to talk about for 1 hour or more. Yes, typically you record neurons which are up to 60, 80, maybe 100 micrometers away from the electrode. And typically you see on each electrode a mixture of a number of neurons. And for this, you do what is called spike sorting, because although action potentials always have the same shape and height, still when you recover, they are sent off from a neuron which is further apart, the signal becomes a bit smoothed and different and smaller until it reaches the electrode. And this effect you use to separate the different spikes. It's a not nice business, prone to a lot of errors. A lot of people thought they could solve it, but it's a nightmare. Even if people sort the same data twice, they get a different result. She's nodding her head. You probably have experience with spike sorting.
I have colleagues with.
So I am assuming we have done all this. I'm not considering issues, what could happen through spike sorting or artifacts or other issues. Then I could talk yet another hour, yeah? So, okay, now I assume I have them sorted out and I have them reported. But for now, even going a step forward, backwards, I would say, when I came into neuroscience, this was, as I said, in the 90s, the possibilities were to record from a very few neurons with a very few electrodes, maybe 2 or 3 or 4. And let's consider we have just recorded 2 of them. Yeah, and we were lucky to record the 2 which are part of the synchronous package. Yeah. So during my PhD thesis, I invented a method which should help to detect what we call excess spike synchrony, which is beyond what we would expect by chance. The neurons fire anyway all the time, right? And then you have some probabilities that they fire at the very same time, yeah? This you can compute. And what I'm doing is that I'm comparing the empirical counts of synchronous activity to that what I can calculate how much I would expect given the firing rate. Yeah, you can do this. And if the empirical counts are higher, then you can add a significance evaluation and say, if this is significantly more than expected by chance, this is more synchrony than expected by chance, and it's an expression of processing. Okay, I give you an example. So this is from an experiment with a monkey where he was first trained to make a movement to a particular target after he had waited 600 or 1200 milliseconds. This was the first part of the experiment. You see here on the left side, you see one of these parts when what he learned. So you see here on top from the two neurons, you see the firing rates as a function of time. You see here the time, and you see the spiking activity of neuron 1 and neuron 2 across multiple trials. And you see here at PS, he got a so-called preparatory signal that he soon will have to move. And after 600 or after, what was it, after 1200 milliseconds, he got the signal to go. Now, funnily, and this we found repetitively, and this is motor cortex, that at the time when the monkey expected the go signal to come but did not, suddenly synchronous activity is coming up. You see it also here in this price function, which indicates the significance. 2 is 99% level, and you see that shortly in time, there is excess spike synchrony. And then it's going back to baseline level. Now, they retrained the monkey to make the movement either after 900 or 1200 milliseconds. And they recorded during the training on what they found at in the second half of the retraining with the new timing. The occurrence of this excess spike synchrony occurred at the new expected signal. So the network learned that he had to expect the signal, the go signal at a later time. So two things, what I wanted to tell you here is this excess synchrony is dynamically occurring, and it is also learned. Yeah. Now let's go a little bit further. Nowadays, these are typically the kind of signals we have to deal with. This also from motor cortex, now with a Utah array of 10 by 10 electrodes. You see the spiking activity of a bit more of 100 neurons. When this increase of the firing rate this is when the monkey moves the arm forward and backward. However, what I want you to look at is that the activities of the individual neurons, these are neurons, not trials. This is a single trials, one after each other. And what you notice is that these neurons do not have the same firing rate. And this firing rate is changing all the time. Yeah. And this is what you want to do in an experiment. The firing rate should change. They should respond, the neurons. This is still the paradigm. However, for me as an, let's call me statistician, it's a nightmare because you have to account for this if you want to go beyond, what is beyond expectation, beyond chance. Furthermore, if you have so many neurons, this unitary event analysis I developed for 2 or a few neurons recorded in parallel. How can we scale this up? And if you think about the number of parameters, if you would go pairwise with 100 neurons, you have 10 to the power of 30 parameters. This is terrible. We have to deal with this somehow. Yeah. And we have, if we would test for each individual pair, we would have a massive multiple testing problem, which would leave us a lot of false positives. Okay, what to do? So we developed new methods. One of them is one which includes as a pre-processing so-called frequent item set mining. Some of you may have heard of this. This is a mining method which has very fast implementations. And we were able to transfer this from a market basket analysis. People are interested what are the people buying in the supermarket together? Wine and cheese, wine and bread. And this kind of analysis people do with what people buy in the supermarket. And we translated this to analyze parallel spike trends. It was a bit of an issue, but we made this so, and now we have a method which is very effectively counting synchronous spike patterns very quickly. Yeah, this is only then at the very end we get some counts of this pattern and of this pattern and of this combination and so on and so forth. So how do we know which one are relevant? And there are typically many. Yeah. So we developed a method which we call SPADE, with the idea to pool some of the results and then reduce the number of tests we have to apply. What you see here is what we typically do. We simulate the data ourselves. These are 100 parallel ensemble processes with the same firing rate. And to test if the method is working properly, we inserted some synchronous events in 10 of these neurons. If I would randomize the neuron IDs for the display, you wouldn't notice anything. You wouldn't see this. Yeah. So it's a needle in the haystack. Okay, what do? We started out with, as before, with a unitary analysis. We count the number of occurrences per pattern, synchronous pattern. And we, on the other hand, we separate this in the size of the patterns, meaning how many neurons are involved in a pattern. Yeah? If these were 4 neurons, then the size would be 4. If this would occur 10 times, then it would be ended somehow with a count somewhere here. And this is what you get from such a data set. And now the question is still, what are all valid or which is important? And what we started to do already many years ago, because the analytical treatment to calculate this null hypothesis of independence of spike trains is basically not possible, we started to use what we call surrogate data. Surrogate data are, you take the original data, in this case, the spike trains, and you manipulate them such that you keep most of the properties the spike train has, but destroy the potential correlation. So in this case, the simplest is, so to say, to touch each individual spike and to displace it a little bit, let's say by plus minus 25 milliseconds or so, if you are interested in coincident synchronous spikes of 5 milliseconds in precision. Yeah, this you can do and hope for the best that all the other features like the interval, interspike interval, and the non-stationarity of the firing rates and whatever you have is approximately the same. Yeah. We made studies a lot on the surrogates. I will not talk about this, but it's an issue for itself to have the appropriate surrogate. For here, we deal with this. We dither the individual spike trains, and then we evaluate these surrogate spike trains the same way as the original data, and we do this multiple times, 10,000 times, and also build up such a spectrum. Yeah, of course, you have to count the same things and so on. And then you notice, oha, all these dark entries and gray entries are actually by chance. Yeah, if you have that many neurons, you have many possibilities to get synchronous spikes. Yeah. Okay, and then we compare the empirical one with the surrogates, which if you have enough, you can express this surrogate pattern spectrum with a p-value as a p-value, and you can directly take out now the individual pins of this pattern spectrum. I inserted one pattern type of 10. And here we see 4 pins which seem to be significant. They are parts of subparts of this big pattern because, but the right one which we inserted is this one. So you see there is still an issue which we have to filter on and so on, but we do. Just want to show you again the workflow of this whole analysis. We have original data, spike data. What we do is we bin them into exclusive bins, let's say 5 milliseconds. We clip the contents to binary values, zeros and ones. Onto this, we apply the FIM. We have pattern counts, we have the pattern spectrum. And maybe we have to correct for some spurious patterns. On the other hand, we destroy the correlation by surrogates, as I said. We do many surrogates. We do the same analysis, binning, clipping, FIM, pattern counts on the surrogate data. We have a p-value spectrum. We compare the two, and we get significant patterns. Just that you see this in one thing. Yeah. Okay. Now we applied this method to experimental data from also a monkey doing a task where he got now also motor cortex recordings. He got first the signal. He initiates the trial himself. Then he got a signal that he wakes up to be attentive. Then he gets a cue, which is composed of 2 dots, 2 LEDs lit up on the right or on the left. And on the right, it means afterwards you need to make a precision task. Or the other one, when it is on the left, you need to make a side trip. Then he has to wait for 1,000 milliseconds, and then he gets the go signal combined again with information about how he has to pull an object. Either then the 2 LEDs on top or on the bottom with a high pull with high force or pull with low force. Then at some point, he leaves the center key, meaning this is the reaction time. And then he does the grasping and gets a reward. Now we wanted to analyze this for spatial temporal patterns, And because the analysis is numerically quite painful, we did not make a sliding window analysis as before, but we cut the data into pieces of same length around relevant behavioral events. And so we had 6 of these epochs, which we concatenated across same type of trials. And in totally, we have then 24 data sets per session. And we analyze this. Now I want to skip this part, but I want to continue, which is similar, same experiment. And want to use, we extended the unitary event analysis to the state analysis, not only to synchronous analysis, so detecting synchronous events, but spatiotemporal patterns. So what do I mean by this? That I have spike constellations where one neuron is firing and then another one sometime data t later, and then yet another neuron firing, yet a delta t later, and so on. And with a total duration of d. And with this exact timing repeating several times. Why? Because these are, so to say, the subsampled neurons from the different parts of the assembly, and we hypothesize that they express spatiotemporal patterns. Okay, so again, with this was not a big change to the whole analysis. Again, this is the same approach. And then we get these examples of these STPs that you get an idea on which granularity we are working. Yeah. So here you see, for example, pairs. What you see here, so you don't see all the 100 neurons, but you see the ones which are involved in patterns in one session. So these are the 6 neurons. And they, for example, exhibit pairs of neurons, pair patterns. Yeah, so we really mark the individual spike. Right? And here you see from another session triplets or quadruplets. And what you also see when I show this is that this is a typical finding when we have about recordings from 100 or 150 neurons or so, only about 5 to maximum 10% of the neurons seem to be involved in this. I don't know why, but this is what we consistently find. So, and here you see, for example, across different recording sessions, each recording session was 15 minutes. You see when the patterns occur during the different periods in the different behavioral contexts, the number of patterns which occurred in there in total, and how many neurons are involved in total in these patterns. This is what I just told you. There are not so many neurons involved in these excess patterns. And the patterns we found were in a range of up to 5 spikes. Yeah, it's pretty warm in here.
Yes. One question. Is it possible to know the source of the synchronity? Like, for example, depending on which pattern you can find, maybe they are electrically coupled with the junction, or they have the same input, or one excites the other. Like, you can know the source of the synchronity depending on the pattern.
That's the problem. No, we can't. There are a lot of works where people try, so to say, so theoretical work where people try to derive the connectivity from the correlation. But this is an ill-posed problem. You cannot solve this. There are too many options and indirect connections and so on. So I go a little bit a different way. I will show you this at the end or later on, that I compare or apply my methods to networks which I know, yeah, and try to find out what the mechanisms therein are, yeah. So, and another issue, what we found, which I found funny, is what you first of all see on the left side. This is what we call it a flower plot of the patterns occurring in a session And these are supposed to be the neurons. Yeah. So for example, these 3 neurons make a pattern, and these 3 neurons make a pattern, and these 3 neurons make a pattern, and these 3 neurons make a pattern, but these 2 make also a pair, and these make also a pair, and these 5 also make a pattern. And what we noticed is that some of the neurons occur in the different patterns. Actually, this was interesting for me because some of you may know the name of Wolf Singer. I was for some time in his lab and was also influenced by him. He had this hypothesis that neurons multiplex their assembly membership. He formulated it in the context of oscillations. But I found this a very sexy idea because you have the time, and during the time, a neuron can participate in this assembly and in another one. Yeah, this is he meant by multiplexing. You know probably the technique from electronics and so on. And I think that this is going on here because these were not occurring at the very same time, but at different points in time. Yeah. And we evaluated this, we call this then multiplicity, that when a neuron is participating, for example, this is occurring in 1, 2, 3, 4, 5, 6 patterns, yeah, and so on. And this also gives us then again a measure which we can use when we evaluate neuronal networks to see whether there are similarities or not. Okay. Another question which some of you may also have had, we looked at this. I wanted to know, are these neurons which are involved in patterns close by? And what you see here is, this is a Utah array, a Utah array, and the electrodes. The X's are not connected. And you see these reddish circles, which indicate which neurons were involved in a pattern in a session. And if the radius is bigger, this neuron was involved in multiple patterns. And what you can see already is that there is no clustering in space, because this would be cortical space, so to say. Yeah. Furthermore, we looked at the distance of these neurons, which you can calculate the physical distance, right? And then relate this to the pattern legs from one of the spikes in the pattern to the other. With the idea behind is this a trivial delay. It's a propagation, and there is just a delay. But what you see here, this is the statistics of many sessions. You see here no correlation whatsoever. Yeah. So these distances are not due to just simple delays between one neuron and the next, but there must be involved an active process. Okay. Okay, now let's go to a model. And I was always searching for, I tell you what I was interested. So in the neurosciences, there was pretty early on, somebody, Moshe Abeles, developed a method, a model, which explains the use of so high connectivity, convergence, divergence. And he proposed that there are layers of neurons, only connections from one group to the next. This is a functional ordering, not an anatomical ordering. And these neurons in the extreme case can be, so to say, all-to-all connected from one group to the next. And what happens in such a network is the following. if you record from these neurons group by group, and you have multiple neurons here in one of these boxes, again, multiple neurons, one group, next group, and so on, what you observe that their spiking activity synchronizes by its own. Yeah, when you stimulate the first group all together, they all exhibit a spike more or less synchronously. They converge on the next neuron synchronously. That's not about me, right? Synchronously. Hello? Okay. So what I want to say is, if you elicit a strong pulse at the beginning, then what happens is that there is a package of synchronous spiking propagating through the network. Even if you dilute this connectivity and not an all-to-all, this is a very strong phenomena. And the reason is because of this convergence, yeah? Because individual neurons, typically also in cortex, have very small EPSPs in contrast to the threshold. You need many neurons synchronously to get across the threshold. And this is, this people call a coincidence detector. You may have heard about it. And this is, so to say, the effect here. You have more or less through the connectivity automatically synchronous activity, and you hardly can avoid such a propagation of synchronous activity. Now, I had the question. I looked at such an AI connectivity, artificial neural network, and I looked at this. This is on the left, and I thought, this looks very much alike, isn't it? You have, I know there are now more complicated connections and whatever, what people make use of, but I don't know why. But the simplest version is that you have one layer after the other connected just between the two layers, right? And they, however, typically AI networks use analog signals, firing rates or so, yeah. However, when our SynFire chain is working with bytes, then I thought I talked to a lot of people who do AI, and then I found a person who does this with spiking activity. They don't do it with spiking because of the learning rule. They want to have some differentiable loss function that you can know in which direction the backpropagation with the errors should go to. This was that people told me this is the reason why people don't do it. Okay, but then I found somebody else. And this is work by the group of Mihai Petrovici in Bern. And this is their first paper on a deep spiking network, which they trained to classify MNIST. MNIST is something very simple. These are these digits, handwritten digits from 0 to 9. Yeah, and you see if they are extremely variable if you look at them, and they have many samples of them, and many ones and many twos and so on. And what they trained is a simple network to with one layer only to classify these digits. And they had two components which I found highly interesting. First of all, they used bytes. Second, these images were latency encoded. If you think of such an image, they are then presented as a, I don't remember exactly, 24 by 24 or 28 by 28 pixels. Then you have darker pixels and lighter pixels. And what I did in the encoding was that lighter pixels elicited later a spike, and the darker ones earlier in the first layer. Yeah, this was the encoding phase, and they trained the network that they had 10 output neurons from 0 to 9, and they trained it such that the neuron which is representing or is supposed to classify the 4 is spiking first. And they made a very interesting derivation here in their work, how they could formulate it with on the level of the membrane potential, But then they had, so to say, because they were able to reduce or lead it back to the membrane potential, in changes in membrane potential, they were able to, so to say, formulate this loss function, which they used for standard error rate propagation. Yeah. I heard a talk of him here a few years, 2-3 years ago here, and I approached him. I said, this was very interesting. I want to analyze your networks. Yeah. I want to encourage you, if you have interest in something, go for it. Talk to the people. Yeah. Otherwise, you don't. I mean, it takes much too much time to do it yourself and so on, but it's there, and maybe the people want to work with you. Yeah. So I approached him and said, Mihai, I want to study more this network. And he, in collaboration with us, he set up a network which has even more layers and also is composed of inhibitory, the blue ones, and excitatory neurons. So the typical relation which is used in neuronal networks, about 20% inhibitory and 80% excitatory. Okay. And then they trained it with this MNIST data set. Now, first of all, I want to show you the spiking activity in these networks when there is a 0 presented. First of all, I want to lead you through one of these. So this is the input, and you see here that the dark pixels elicit very early a spike, and the lighter the pixels, the later a spike. You see this activity in the first layer of the excitatory neurons with some pulse, the inhibitory ones. And this is then going through the network, and at the very end, you see a very slim synchronous package just before the output neuron, which is eliciting then the correct output neuron. Can you see this? And if you look at this for the different digits, it looks alike. Activity looks alike. So I thought, how should this go? I mean, how does this work? And then we did the following. We were first looking at which neurons fire to which input. Now, you also need to read this. This I ignore for now. This is the input. But for example, in layer 1, you see here multiple presentations of a 0. This is this first box. And then you see here, this is the axis of the neuron IDs. And you can see which neurons are active for presentation of a 0. I know it's a bit dense, but if you go across this, you notice that the seemingly random dots become more and more like lines. You see this? That in the layer 4, for example, almost the same neurons are firing for the presentation of a 0. Right? See this? This kind of barcode-like thingy, yeah? The below ones are the inhibitory neurons. They fire in tendency a bit more, but it seems to be not so clear here, yeah? Okay, this was what do the individual neurons do. But now we look at, so to say, at the joint activity across the neurons. So we looked at these vectors, for example, we wanted to compare, are the same set of neurons active for when a zero is presented, different zeros are presented and correlated or computed the similarity of them. So this in this case is from a 0 and a 1 compared, so different labels. And you see here the correlation coefficient of these. And you see, for example, that this coefficient is lower when you compare different responses to different labels as compared if you use the response from 2 sixes, for example. It's typically higher, and it becomes, this distance becomes larger and larger. They become more specific, yeah, through the layers. Now, we also, that's the nice thing. If you have such a network, you know everything. You know the connectivity, you know the weights, you know the activity. It's really great. You can do whatever you want, which I typically cannot do. Now, this is the weights. So what we also found out is, so I was talking about the activity. Now I'm looking at the connectivity and the weights. And what we found is that there are specific neurons having strong weights to the next layer, to particular neurons for the different output neurons. It's kind of logic somehow, yeah, but I wanted to see this. So this is, so to say, the basis that particular neurons, different neurons, are firing for the different digits. Yeah, they go a particular path through this network to the 3 as compared to the 7. And how is it with the activity? Well, this is exactly the same thing. The activity is, first of all, like in a synfire chain, synchronizing more and more through the network. These are different sets of neurons which are propagating through this network. And the funny thing is, we looked then into a book of Moshe Abeles of 1982, and he had in the very last chapter, he had this drawing. Also I added, but he had this drawing. So what he was talking about, that you could have multiple first overlapping synfire chains. However, they separate more and more, that they become different paths which in our case converge to different labels. So I was super happy about this finding. Here is a preprint. It's currently under review. It will be soon, I guess it will soon appear in physical review. I don't know. Okay. Overlapping synfire chains as predicted by Abeles. So he started out with theoretical considerations of the brain and because of the connectivity. And we found this in a AI network. Yes, please.
Maybe you can understand that how the weights evolve in this network, like the synaptic plasticity, how it evolves the weight. How we
didn't follow this, but we are currently doing this. But what I heard from our colleagues, that I didn't know that these weights are not so much changing over time, actually, as if you would make use of a certain bias in the system anyway. Yeah. How they exactly evolve, I don't know, but they said they change only very little. Okay, I'm coming to my end. Neurons compute by interaction. They interact by coordinated activity. This is expressed in correlations. The signatures are, for example, spatial or temporal spike patterns. Their detection suffers from subsampling. Still, we found patterns in relation to behavior, and spiking deep networks use the same mechanism. And I want to thank the people working with me and my funding, and want to thank you very much. But there were already questions in between, so.
Yeah, so if there is still some.
Yeah, I mean, I have one question like this. First is I really enjoyed your lesson. The question is, now are you trying to subsample the same spiking neural network that you are using in order to try to correlate if what you find in the laboratory makes sense to this?
It's on my list. It's very recent, but very likely it's a bit more difficult because we noticed that for different digits, for different zeros, the speed of running through the network is different. So it's a bit difficult to find identically repeating patterns.
I think that the pattern worked with simplicity or so, that was 5 to 10%, but you check that this is a kind of subset thing, so if you're not working with the for instance, elements of data, would you guess there's no.
Good point. That's a very good point. I'm currently exploring more of artificial networks to also get at this point, but or whether it's also a flaw in the analysis, because this frequent item set mining on binned data may also consider, how should I say, chance patterns at some point, because yeah, and so on. I'm working on this, but it's very good that you have so many ideas that I can continue working. More questions?
Yeah. Question from my slides. Do you think that this framework also doing this type of analysis can be, how do you see it becoming useful in the future also to help us understand situations where there is a medical condition going on and we want to understand better information processing in networks and to try to create better types of therapeutic design, and very much looking to the future, of course, because we're still trying to figure out
Yeah,
patterns. But
I'm not so much into this, and typically the medical people don't look at the level of spikes, right? However, I was talking for some time to Angelika Lampert, who uses MEAS, so she has growing tissue and 4,000 or more electrodes, and wanted to know how to analyze this. So in that sense, we are in contact with people who want to use this. Yeah.
I think that's also these stimulation devices that are becoming now more in development. They could take into consideration some kind of disinformation, alterations at some point when we master them a little bit more, no? Hopefully.
Yeah, I don't know if ever we want to activate individual neurons, but maybe this can be done, I don't know. But this brutal, sorry, Peter, brutal TMS stimulation. This is not my thing, yeah. This is, so to say, areas activating, and this is very, for my sense, a bit unspecific, but you can comment on this.
Okay. So if there are no other questions, thank you again. Time for the
So thanks for the invitation and the kind introduction. I'm sorry that I can't be there today in person. It's just like a scheduling conflict that unfortunately wasn't avoidable. But for the duration of the talk, like if you do have questions, please ask away. Like I actually prefer if people are asking questions during the talk when I do remote, because otherwise I'm just sitting here talking to my computer. And after a while, that becomes very weird. So if I get questions, I realize that someone is listening, and I'm happy. So yeah, so feel free. In the presentation, what I wanted to do was to give you an overview of the different experimental approaches that we're using to study neural brain function. And we're focusing our experiments on a mouse model where we study the brains of mice that are performing different behavioral paradigms to understand how they process sensory information and how they turn this information into behavioral output. And yeah, so that's the general gist of it. Let me just quickly see if I can hide my Zoom bar so that it's a bit easier. That did not work. Okay, you still see the screen, right?
yeah.
Yes.
Okay, so now I don't see you anymore. So yeah, just speak up if you have a question. Okay, so as I mentioned, the general focus of the lab is to basically study how the brain generates behavior. And the general approach that we're taking is that we look at a sensory input and then basically ask how the brain processes that sensory input to produce a corresponding behavioral response. And so one thing I'm noticing right now is that these slides are really slow. Should have tested this before. Let me quickly see if I can understand why. I think my network is not great. Okay, but the point is basically that we want to study how sensory inputs are being transformed into meaningful behavioral responses and what happens in between. And the brain structure that we focus the most on is the neocortex, which is the outer shell of the brain, which is parcellated into functionally specialized subregions. And in very broad strokes, you can separate them out into sensory, association, motor, and executive areas. And the basic idea is that as the information enters the neocortex, it's being transformed from raw sensory information into an actual behavior decision that will translate into behavior. And as I mentioned, we use the mouse model as our model of choice. One of the reasons why we like using mice is because we have this very large toolbox for studying neural activity. So one of the things that we do is we use this method called wide field imaging, which we can perform through the skull of mice that are performing a behavioral task and observe the entire neural activity across the cortical surface as the animals are performing these behaviors, which is really neat if we want to study the interaction between these different brain regions. So this is an example mouse that is sitting in a behavioral setup. And at the same time as the animal sitting there, we're looking down onto the brain surface of the animal to study the neural activity. So what we see here is a top-down view on the animal's brain. So you can see the cortical surface, and you see all the brain vessels that are innervating the cortex here. And when we overlay this field of view with an atlas of the mouse brain, where we can see the location of different brain regions, you can appreciate that with this field of view, we basically see the entire cortical surface. So the reason why we see anything in this picture is because these mice are transgenic animals. And what they're doing is that they express a calcium indicator. So that's a protein that binds calcium and then basically translates this binding into a fluorescence signal that we can measure with the camera system. So what we see here is the fluorescence that comes from this calcium indicator. And if we want to measure neural activity, all we need to do is to basically measure changes in fluorescence, right? So that's our proxy for neural activity. In these mice, that calcium indicator is expressed in all excitatory neurons. So basically what we see here is the activity of the cortex in the neurons that are exciting other neurons. And so this is how this looks like. So you can see on the left, the animal is doing a behavioral task. And as the animal is performing this task, we see pretty pronounced changes in cortical activity that are linked to different behavioral patterns. And interesting enough that this is not just limited to the task itself, but to any type of behavior that the animal decides to do. So as it starts grooming itself, for example, you see that drives a very distinct cortical activity pattern. And when it stops doing so, we see other activity patterns. For example, when it uses its forepaws, get these very distinct activity patterns. So the behavior that we use now to actually study perception it takes many different shapes and forms, but the basic design that we use is what's called an evidence accumulation task, where we present stimuli to the left and the right-hand side of the animal, and the animal needs to learn to make a behavioral response on the side where the stimulus was presented. So in the simplest form, there's only a stimulus on the target side, and the animal then goes to that side and gets the reward. Was there a question, or I heard a weird thing? Okay, well, apparently not. You still hear me though, right? Again, I see literally nothing. I'm just talking to my slides right now.
Yes, we
Okay. Yeah, so maybe this was on my end. I don't know. It was weird sound. Okay. But the point is basically first you train the animals to just report on which side they perceive the stimulus. But once they can do that, you can also start presenting stimuli on the non-target side. So in this case, this is an auditory version of the task where we present click sounds on both sides, and the animal then basically needs to accumulate the overall amount of click sounds that were presented and differentiate between the target and non-target by choosing the side where more clicks were presented in total. So in that case, they really have to accumulate this information over time. And then in addition, we usually present a delay period where the animal have to hold the decision in memory for a moment. So we separate the perception of the stimulus from the formation of a choice from the actual motor output that is created when they make their response. So they can do this really well. So this is an example of learning curves from 21 mice that were trained in this auditory behavior. And you can see it usually takes about 10 days until they reach decent performance in detecting that stimulus side. So that's usually about 2 weeks of training until we have animals that do really well. And you can also see that they reach really high behavioral accuracy in these tasks. So what I'm showing here is an example where we use both auditory and tactile stimulation. And you can see the amount of stimuli that are being presented on the right-hand side on the x-axis. So that's here. And then the amount of times that the animal actually chooses to go to the right as the actual response. So that's shown here on the y-axis. You can see that there's a direct relationship between the amount of stimuli on the right and the chance that the animal will actually choose this as its target. And when you plot it like this, this is called a psychophysical response curve. So it gives you a sense for how well the animals can do this discrimination behavior. Okay, so to give you a better idea of how that could look like in practice, here I'm showing another video angle. So this is looking at the animal from the bottom, and you can see the animal's belly and the bottom side of its face. And in the baseline of the task, the animal's just sitting stationarily, and then we have these handles that can be moved closer to the animal. And when the animal's touching these handles, in this case, it would indicate the start of a trial. Like this is not all our behavior paradigms use this, but it's a feature we can use to allow the animal to indicate when it wants to start the stimulus presentation. And then there's the stimulus period. So in this case, this is a visual trial because it's easier to show in a video. But you can see there's a stimulus on the right-hand side of the animal. And now there's this additional delay where it just has to wait. And then when it comes to the response period, you can see these two spouts that are in front of the animal. So these are two little water tubes. And in the response period, we're moving them closer with motors. And that allows the animal to then make a response by just licking on the side where it thinks the stimulus was presented. And if that choice was correct, it gets a reward, and then the spouts move out again, and the next trial starts over. So this is just an example for one of these behavioral paradigms that we're using to basically probe the animal's perception and its ability to make choices based on that input. Okay, so when we look at the brain now, how do we actually study neural activity in these behaving animals? So one thing that's really interesting about imaging is that you can actually express that calcium indicator that we use as our readout in specific subsets of neurons. So one thing that we've been doing was to target the expression of the calcium indicator to specific neurons, in this case called pyramidal tract neurons, which are sitting in the deeper layers of the cortex. So they're primarily sitting in layer 5, which is kind of deemed to be an output layer of the cortex. And these neurons are sending projections to many different subcortical regions. So you can see this here where the neuron sits here in the cortex, but then it innervates the striatum, the thalamus, the midbrain, the pons, and even the spinal cord. So these neurons can form these very long-ranging projections to subcortical areas. In a different mouse line, we express the calcium indicator in a different class of neurons. So these are called intratelencephalic neurons. And in contrast to the pyramidal tract neurons, these neurons are primarily forming connections within the cortex. So these are not output neurons, if you will, but they are more intracortical communication neurons, right? So those could be the kind of cells that allow different cortical areas to communicate with one another, or also to support local computations. And aside from the cortex, IT neurons also project to the striatum for developmental reasons. Okay, is that clear so far?
Yes.
Okay, so because we have the calcium indicator in these neurons, we can now start using the wide field imaging to study these cortex-wide neural activity patterns specifically from either pyramidal tract or IT neurons, which are both excitatory neurons. But one important question was if they're actually performing different computations because of their difference in genetic identity and anatomical projections. So this is just to show an example of how that looks like in practice. So here we see these cortex-wide activity maps, either from a mouse line that expresses the calcium indicator in all neurons. So that's the one on the left, it's called EMX, or then either from the IT or the PT neuron specifically. And this is just ongoing activity. So the only thing I wanted to show with this videos is that it works really well, right? So in all of these mouse lines, we get these cortex-wide activity patterns, and we also have really high signal-to-noise ratio, meaning that even on a single trial level, it's very easy to see which regions are being activated. Okay, cool. So one thing we can do now is we can look at the cortical activity during different moments in the trial, right? So I told you earlier that the behavioral paradigm has these different stages in the trial. So we can now look at the average activity in the wide field, for example, when the animal initiates a stimulus or during the stimulus period itself, or then in the subsequent delayed period, or then here in the response period. And one thing you can see from these maps is that in the different mouse lines, we do get different activity patterns. So this was already an indication that if you have this cell type specific imaging, you indeed see different types of communication, even in the same areas. However, unfortunately, these are really complicated signals. So when we got this data, it was a bit tricky to look at them and go, aha. But it was more like, okay, like why is the activity so low during the stimulus period, and what's happening during the stimulus, like what's happening during the delay, and like in all these kind of traces, you can see that it's not exactly easy to see the signature of a specific process because they're all happening at the same time. So in this case, for example, there is a activation of the auditory cortex here, but that activation happens even before the stimulus. So the stimulus here is shown in gray. And so in this case, this dashed line shows us the moment when they initiate. And that initiation drives strong activity in the auditory cortex, as well as the frontal cortex. It's pretty widely distributed, actually. And then after this activation, the activity goes below the baseline, right? So you can see how the activity dives down here. And then maybe there's a stimulus response. Like, you know, there's like this peak here, but it's really tricky to like be sure that we can accurately assign what these different activity profiles actually correspond to. And similarly, when we look, for example, during the delay period in the frontal cortex, you can see how this activity is ramping up here, and maybe that is a signature of the animal making a choice. But again, it's far from obvious how we should look at this kind of data. So in order to deal with the complexity of this wide field data, we use a linear regression model that allows us to basically assign different parts of the activity to different types of events that are happening during the task or that are driven by different types of movements, for example. So, for example, in a given trial, we know that the animal, for example, chose the left-hand side, and there was a stimulus on the left side, so that makes sense. And then we also can look at the movements that the animal was doing. So, for example, we can look at these licking events. You can see it was licking on the left more so than on the right. And then also here we can see if it touched these handles at different moments during the trial. So these we call instructed movements because they're directly linked to the task, right? So the kind of movements that you would expect if you want the animal to do the task at all. However, on the other hand, there are many other types of movements that are uninstructed, right? So for example, if you remember this grooming behavior that we saw earlier in the video, that kind of behavior hasn't been kind of asked for, but they naturally happen because these are animals and they're doing all kinds of stuff as we run our experiment. So in order to also account for these uninstructed behaviors, we use, for example, the video data that we have And in that example, we extract the motion of the whisker pad. So in this case, we compute the motion energy in the whisker pad, and then we threshold it to basically get these events when they're using their whiskers. So the whiskers are these vibrissae that they have in their face, which they like to use to basically get immediate touch around their face. And once we have all of these events, we can then build what's called a design matrix, where we basically try to build a little matrix here of these different regressors. So this is when the event actually happens. And we can then basically shift them forward and backward in time to build a large matrix of regressors that sample different time points around the occurrence of these different events. And once we have this regressor matrix called the design matrix, we then can basically build a regression model to predict the neural activity based on the occurrence of these different events. So this is something that's called rich regression. So rich regression is basically just a flavor of a regression model. I think that's more technical than I want to be, but in case you have questions about what that means, feel free to ask. But the point is basically that we use all these different events to then predict changes in the neural activity. And we basically integrate the task, the instructed and the uninstructed movement all together in one large model that is doing this prediction. And one thing that's nice about it is that because we have these combination of different regressors that are tied to a specific event, we can then ask basically how that event specifically contributes to the prediction of that model. So the idea is basically first you build a model that does a good job at predicting the neural activity. And once you have this good model, you can look at the inside and basically ask which components are responsible for specific types of responses. So in this case, this would be what we call a response kernel. So this is the regressors that are tied to the stimulus variable. And one thing that I think you can appreciate even at this level, that in that case, there's much more structure that is tightly linked to the stimulus compared to what we've seen before. Or at least it's obvious to me, but I've been looking at this for a long time. Okay, so once we have these different kernels, as I mentioned, we can explore which parts of the cortex are activated by different types of events. And they're not limited to the ones that are part of the task. So in this case, for example, this is the response kernel that is tied to the animal touching the right handle. And you can see that there's this very specific activation in the left hemisphere of the cortex in this region, like this little bell-shaped kind of thing. So I did a poor job here. But this area is exactly where you would expect the somatosensory forepaw region to be. So this is somatosensory cortex, and then we also have primary motor cortex right next to it. So this is highly spatially specific. And at the same time, when we look at the response kernel to the auditory stimulus, here we see that there is very clear activation in this very lateral region here on the side, and that's the auditory cortex. And then we see activation here in this region, which is very close to that somatosensory area that we saw with the handle. And then there's also activation right here in this frontal cortical region. So in that case, basically we isolate. So this is part of this area called parietal cortex. And we basically see this chain now from auditory to parietal and then frontal cortex, specifically tied to the stimulus presentation. Okay, so once we have done that, we can now, as I mentioned, we can explore this further. And here we're just showing the temporal dynamics of this response kernel. But we can then also compare these responses to the different mouse lines that we had in the experiment. So we can ask how the pyramidal tract or the intraencephalic mice look like in comparison to this imaging that is just using all the excitatory cells. And what we found here is that they still look really different, right? So one of the major findings here was to show that when you have the cell type specific imaging, you get really different response maps. And we still see that there's activation in the auditory, parietal, and frontal regions. But when we compare the activity between these two mouse lines, the PT and the IT mice, we can see, for example, that in the parietal cortex, so here I'm just zooming in on that part of the cortex, you can see that it's really distinct parts of the cortex that are being activated. So in the IT mice, for example, we can see that there's a hotspot in this region, but in the PT mice, it's more shifted medially, so like towards the right, which is towards the midline. Right, and you actually see that it doesn't really respect the boundaries of this cortical areas anymore. It's between different regions in this case. And then in the IT mice, we also see that there's a secondary activation that is more in the front, which we don't get when we look at the pyramidal tract neurons. Okay, so this was looking at responses to a stimulus. But how about the part of the activity when the animal is actually turning that stimulus into choices? So here I wanted to show a different type of analysis, which is related to the linear model approach, but it's using a choice decoder. So what this type of analysis means is that you, instead of using the events that you can observe to predict the cortical activity, you use the cortical activity to predict a specific variable of interest. So in that case, we use the cortical activity to predict what the animal will do, right? So we want to predict its choices. And first of all, when we do an analysis like this, here we see the accuracy of a choice decoder against time. And you can see that this choice decoder is actually pretty good at predicting what the animal will do, even before the presentation of the stimulus. So one thing that's interesting is that basically even in the baseline period where the animal has no information about the stimulus at all, will actually do a decent job at predicting where it will go, which also means that many of its choices are not related to the stimulus because the animal already made up its mind. Like there's already a signature of the cortex as to what the animal will do, even before it does what we would then call the right thing. The other thing that's interesting here is that you can see that the prediction kind of ramps up as we move through time until we reach this dashed line, which is when the animal actually starts moving and execute its choice. And then the decoder becomes very, very strong because the reflection of this motor activity is super obvious in the cortical data. Okay, any questions so far? Is that everything makes sense? Okay, so this decoder also gives us weights. So these weights can be interpreted in a similar way to what we get from regression models. In that case, this prediction just is a bit different in terms of how do you interpret it, because it's not really the change in neural activity itself. It's the weight that you put on the neural activity to do a good job in predicting the choice, right? So this is a nonlinear decoder approach where this is just logistic regression, but it still means that basically you turn the activity into a binary choice, left or right. And you basically rescale the activity so that it does fit well onto something that you can then threshold and turn it into a binary outcome. But what's interesting about this is that basically these kind of weights are not so dependent on how much activity there is in the cortex, but which signals are the best predictors, right? So this is basically an approach that highlights regions that are particularly sensitive to differences between left versus right choices. And one thing that we saw here was that there's this frontal region here where there's also this dashed circle where you can see how this immediately lights up once the stimulus is being presented. Right, so this is in the initiation phase and there's no signal here, but then once the stimulus comes up, there's very strong choice predictive signals in this frontal area. And this persists right through this delay period until the animal actually makes the response, and then these signals dissipate. Okay, so again, we can take an analysis like this and compare it for the different cell types. And what we found here was that depending on the cell type, we actually see an inversion of the choice specificity. So this was something that's interesting because here we see that in the IT neurons, we actually get a different sign on the choice decoder. So in this case, the activity predicts choices on the same side, whereas usually the activity here would predict choices on the other side. Maybe it's going too deep why, but it's just interesting to say that with the cell type specificity, we get these really different signatures of choice coding in that area. Oh yeah, and there's some details that these signals are also a little different in their spatial extent. Okay, so then once we've done the imaging and we see these different areas that are interesting when it comes to processing sensory information and turning them into choices, there's always this question as to what we saw actually being truly linked to the behavior. Because a general issue is that just because we measured a signal doesn't necessarily mean that it's causal. It could always be that what we see is just correlated to the animal making a choice, but it's not really driving the choices at all. So because of that, what we often do is that we supplement the imaging with optogenetic inactivation. And what that means is that you want to perturb the area that you identified for being important to then causally test whether that activity is indeed linked to the behavior. So the prediction would be that if this is an important area, if we mess with it, the behavior will be affected. And similar to the imaging, you can also express, in this case, an inhibitory opsin to different subtypes of neurons, and then basically, again, ask that question with cell type specificity as to whether a given area is important. So in this case, we use viral techniques. So we inject different viruses that can trigger the expression of these constructs. And once the construct is present in the neuron, if we shine light on that area, we will inactivate the activity of these cells. And the other thing that we can do is that not only are we inactivating a specific region, we can also do it at different points in time. So in this case, we can inactivate either early or late during the stimulus, or we can inactivate during the delay, or during the response period, with the idea that that gives us additional information as to whether these neurons are more involved in sensory processing or in the formation of choices or motor output. Okay, so what do we get when we do something like this? So this is the result for performing this optogenetic inhibition in this parietal cortical region at different points in the trial in a mouse line that is all excitatory neurons. And what we see here is that there's an impairment in the animal's ability to report correct choices, right? So the y-axis here shows how strongly do we affect the animal's ability to give the correct response. And one thing that's interesting here is that the effect is the strongest when we do it early during the auditory presentation. So this goes well with this idea that this parietal region is indeed important for processing auditory information. But then as we move later into the trial, its impact on behavior becomes weaker. And now we can ask for the role of different cell types. And when we look at these pyramidal tract neurons, we actually still see a clear impact on performance when we inactivate them, right? So it's not quite as potent as when you inactivate everything, but the general profile is pretty much aligned to what we saw before. But interestingly, when we now do the inactivation into intratelencephalic neurons, as shown here in green, you can actually see that the effect becomes even weaker, and it's actually not significantly different from zero anymore. Right. So in that case, there's only a very weak trend of that doing anything to the behavior, which was a really surprising result because many theories about the function of this parietal region, for example, are heavily built on the idea that this is a hub region within the cortex that coordinates the activity across cortical regions. Right. So if that's its central role in the task, you would have thought that inactivating these IT neurons would have really done a big impact on the behavior. So instead, the fact that these PT neurons were so important suggests that there's interactions between the cortex and deeper subcortical regions, so areas within the brain that are actually very important for understanding what's going on in this task. And then we move to a different area. So this is moving from the parietal to the small frontal region. And in this case, we get a different profile. So here you can see that the effect becomes strongest right during this delay period. So basically after the stimulus, but before the animal's choice. And in that case, we see that when we inactivate here, the animal is very strongly impaired, which goes well with the idea that this is a region that is important for the formation of choices. Right, so this also explains why there's already an impact during the stimulus, but that impact becomes even more pronounced as you get closer to the animal's response. And we do see similar effects in this case, both in PT and IT neurons. So in that case, it seems that both of these neural populations are important for the correct function of that area. The only difference that we found was that in the response period, the PT neurons seem to be more important than the IT, which suggests that when it comes to motor production, it's primarily subcortical projections and not so much intracortical that are still required. Okay, so this is a short summary of this part of our work. So I showed you some of the cell type specific cortical imaging and that you can see very different types of cortical coding dependent on whether we look at PT, IT. Oh yeah, and there's corticostriatal neurons, but I took that out of the talk for time reasons. And depending on the kind of analyses that you can do, you can look at different features such as stimulus and choice-related activity, and we can analyze them across all these cortical regions. And when it comes to looking at sensory responses, we saw this very pronounced signature in the parietal cortex that highlighted different subregions depending on the cell type. And when it came to choice, we see pronounced differences in the frontal cortex that also is mirrored by these causal perturbations that we did. Okay, any questions on this part? Like trying to get my video back, it's really weird because I managed to really not see anything anymore. Well, I know I'm alone. Okay, so there are no questions, right?
No.
Okay. So I'll just keep going then. Just stop me. Like if things get weird, just tell me I should stop. As I said, I don't see my Zoom anymore. Okay, but focusing more on that part about subcortical projections, another technique that we can do is we can look beyond the cortex by specifically measuring what the cortical neurons are doing, or basically what type of information they're transmitting to subcortical regions. So in this case, we focus on this frontal cortical area, like the one that we identified with wide field imaging, and then we inject in this case, so this is just a dye in this case, where we basically just want to identify where these neurons are actually sending most of their projections to. So in that case, we found that there's 3 specific regions in the brain. So this is called the striatum, the thalamus, and the deep superior colliculus, which is a part of the midbrain, where this area has very strong projections. And this also goes with literature. Like there's a couple of papers that also saw similar results, but we just did this to confirm that we know what we're doing. And once we have this anatomical data, we can now place a glass fiber in a specific in these specific regions to measure the activity of the axonal projections from the cortex to these regions. So the idea here is basically that we still have the calcium indicator. In this case, we actually express it only in that frontal cortical region. But then it's not only in the cell body, it's also in the axons, right? So in the projections of these neurons to these different subcortical areas. And by using the glass fiber, we can then measure the activity of the axons as they're being activated in these different subregions. And the appeal of this approach is that we can basically parcellate what that area is communicating to these different recipient areas. Okay, so when we do this kind of experiment, we used a slightly different task. In that case, we've started off by something that we call an innate task, where the animal basically just has to collect the water. We just give the water either left or right, and the animal just picks it up. So that is more a motor task rather than interpreting sensory information. And then once the animals are doing a good job in licking, we then start by performing this auditory task that I explained before. And then we add the delay to the auditory task. And once the animals are performing well with that, we return to the innate task. And the reason why we do it sequentially in this case is because we were interested in learning. So the idea is basically to ask how these axonal projections change the information that they're transmitting depending on the animal's learning progress in these different tasks. And this innate motor task is basically a nice baseline to also come back to ask how learning changes the type of information that is being transmitted, even when it's not actively required. Okay, so this is the kind of data that we see when we do the photometry. So this is just for the ALM. So in this case, this is still using that glass fiber approach, but this is basically the bulk activity in the frontal cortex. So ALM is this frontal cortical region that I showed you earlier. And you can see that there's some modulation during this innate task. So there's this ramping activity, and then there's a peak when the animal makes a response, but it's fairly stationary. So like there's maybe a jump from the first session to all the subsequent ones, but overall there's not super much happening. But then when we start presenting these auditory stimuli, what we see is that there is a change in how the auditory stimulus drives activity. So this is the dashed line here in the middle. You can see that super well, but basically the blue lines are initially fairly flat. And then only over the course of time, basically, we start seeing these very clear responses to the stimulus here. And you can also see how the activity then towards the end of the stimulus period starts increasing, and the peak during response is also getting much larger. So in that case, we see these very pronounced differences that are a signature of the animal learning this auditory task. However, once it did learn this task, we can then start presenting the delay. And as we start presenting the delay, there is still kind of like additional fluctuations in how this then looks from session to session. And then in the end, we can return back to the innate task that we did before and basically ask whether this learning experience has a strong impact on the neural dynamics. And the one thing that we're currently looking at is you see this negative component here that is basically reducing the activity even before the stimulus is presented. So this is an interesting feature that kind of evolves even here early in the auditory task. You see how the activity is diving down here. That is maintained even when we return back to this innate task. So we treat that as a signature of attention, actually, but it's just really interesting how that's one of the more salient things that are conserved even in this very simple task. However, as I mentioned before, the actual appeal of the photometry is to look what happens with the axonal projections. And this is an example now from recordings of the axonal projections from that frontal area in the striatum, right? So, and one thing that you see pretty quickly is that even in the innate task, actually there's much more variability in the activity across sessions in the striatal projections. Right. So in that case, you can actually see how these different lines basically cover more ground. So there's more evolution. In this case, we even see that negative component even evolving here. And you can also see that these learning related dynamics are much stronger when we compare them to the measurements in the cortex. Interestingly enough, in that case, when we move to the second task, the one with delay, when the animals are more expert, this amount of variability is actually reduced. So it seems like the axonal projections from the frontal cortex are particularly engaged in this early learning phase, like so in this early innate and this auditory task, but then they basically become more stable as the animals turn into experts. Yeah, and then this is an example from the superior colliculus. So same idea. And in this case, we look at the axonal signals in this midbrain region. And again, we see these really pronounced changes across all these different tasks and much stronger signatures of learning compared to what we saw in the cortex itself. And one thing that's interesting here is that, first of all, you see that this is kind of more maintained also in this other task, the task with delay. And we see these very strong modulations in the response, like in this motor activity component that is more pronounced compared to, if I go back here, that is more pronounced compared to the other regions. So yeah, so we're still actively analyzing what this all means. So unfortunately, I can't really give you a nice punchline here, but I thought it's worth mentioning basically that these are the kind of tools that we're currently leveraging to really understand how cortical and subcortical regions share information and the amount of specificity that can be embedded in these different projection pathways, even when they're originating in the same cortical area. Oh yeah, so I forgot about the thalamus, but as I said, this is all very observational. There's other stuff, but I would maybe point to this negative component being particularly pronounced in these data. So if this is a signature of attention, it seems to be particularly important for the interaction with the thalamus. But yeah, I'm not very firm on that just yet. Okay, so once we have this photometry data, we can basically look at it and we can basically try to make theories about their respective importance during learning. But this is ongoing analysis, so if you have questions or suggestions, be my guest. But it's a really nice technique because you can do these measurements in the same animals as well. So one thing that is very helpful is that when we do these photometry measurements, we can also compare them with each other, which is much more informative compared to using cohorts of animals where you need to use more animals and the variability across individuals becomes really problematic. So we can again basically turn this around and then ask causally if what we saw from the imaging is really just a signature of basically like correlative activity, like, you know, like it could be related to learning, but maybe it's not, we don't really know. So in that case, you can also do inactivation of these projection pathways. So in this case, we can use a synaptic silencer to suppress the activation of a specific pathway. So actually what the silencer does is it prevents the release of vesicles. So the axon becomes activated, but the synaptic release is suppressed. So this means that the target area doesn't get to see the actual effect of that activation. And it's the same idea as what we did before. So basically, if we say a specific pathway is important, we can use the synaptic silencer to then basically shut down that pathway specifically. And here's an example for a control experiment where we do a measurement in the striatum. So in this case, so there's an electrode here in the striatum. And then we activate the frontal cortex, and we see that there's a response in the striatum when you activate the frontal cortex. And then when we activate this optogenetic silencer in green here, we can actually see how that response becomes weaker, right? So that peak, because of the silencing of that pathway, is suppressed, and then it returns over the course of about 10 minutes. So there's a quantification of that here, where you can see it goes down and then it comes back up. So this is a really useful tool to basically study the function of specific pathways and really try to understand what a given area communicates to different subcortical regions. And what we found with that approach was that when we do this inactivation of specific pathways, we see the most behavioral effects early in learning, like in this first innate and the auditory task with no delay. But then when we look at the deep SC and the thalamus projections, in that case, these pathways seem to become more important later in training. So there's a nice dichotomy between the importance of these two different pathways depending on the animal's experience, where striatum is important early and then SC and thalamus are more important in late experts. Okay, so this concludes this part. I'm going to stop asking if anyone has questions, but again, like just feel free to shout. The other part I also wanted to talk about was everything I've said so far is based on a specific sensory input driving the behavior response. But in nature, it's usually more than one sensory input that is driving the behavior. It's basically a cacophony of different types of inputs that are all need to be integrated to form some kind of coherent multisensory representation of the world. So this is why we also study what's called multisensory integration, with the idea that we want to know how different inputs are being integrated together to drive behavior. And so for that, we have a different type of task. So this is a multisensory discrimination task. It actually works pretty similar to what I described earlier, but in that case, the stimuli are visual and they're presented either to the left or the right-hand side. And then we can also turn this into a discrimination task, as I mentioned before. But we either have these visual gratings, so this will be the visual part of the task, or we have these little air puffs. So basically, we just puff a bit of air to the whisker pad of the animal to provide a tactile stimulus. So in that case, basically, we can choose whether we want to do the task either visual or tactile. And usually we first train the animals in the visual task. And here you can see a learning curve that is pretty reminiscent to what we saw with the auditory behavior. But once they have learned this task, we can then start using tactile stimulation. And one thing that's interesting here is that they can not only learn the tactile version, they actually learn this task much faster compared to the visual one. So they're able to transfer the knowledge that they had learned from the visual task onto this new modality. And then lastly, when we present both modalities together in a coherent way, we actually see that these multisensory stimuli are producing a stronger response. So, or not stronger response, but higher performance. So what this means is basically that immediately when you present both stimuli together, the animals are able to leverage the fact that they have more information and improve their behavioral performance.
And have you
Yeah.
have you tried instead of first visual and then tactile the other way around?
No, that's a good question. So unfortunately, these experiments take so much time and effort that we didn't get around to it, but that would be really critical to investigate this transfer learning better, right? Because it's possible that they just learn tactile stimuli more easily. Yeah, unfortunately, I don't have the data to address it just yet. But great question.
Thank you.
My guess would be that it can't be the only thing because this learning is just too fast. Like we've had animals that basically learn the tactile behavior immediately within the same session. And that's something that's pretty unheard of.
And do you think it has something to do with the thing that you're blowing air to, like the part of the nose that is nearby the mouth? That's how they lick.
I don't know. I mean, it's possible that it's easier for them to build an association when the stimulus is kind of more localized and it's kind of more spatially linked to where the robot is. It's really hard to tell because guessing what the mouse might experience or what it thinks about inputs is very difficult. However, it's tricky because if we overdo it with the strength of the stimuli, it can easily turn into something like a deterrent. Like, it could be that the animal doesn't want to go where these weird puff thingies are happening. And it could also be that if the air, for example, gets too close to their eye or it's just discomfortable, like if it's not comfortable the way that the stimuli are being presented, this can actually impair their ability as well. So you have to really carefully titrate it. And yeah, so that sometimes makes the interpretation a bit harder because we don't ultimately know what exactly it is that could make it easier or harder for an animal to learn the task. So it's difficult.
Okay, thank you.
Okay, so however, once we persevere and we train these animals in this multisensory task, we can so there's also again a psychophysical curve here just to show that with the multisensory stimuli, their overall performance is enhanced. And we did some follow-up analysis to show that in the multisensory trials, they're basically performing as you can expect on a theoretical level if you just additively combine visual and tactile information. So it's not like there's some magic thing happening, but they're able to independently combine both unisensory streams into a consistent whole. Okay, so now turning to the cortical activity. So we can again use the wide field imaging to look at how the cortex becomes activated. In that case, we look at this delay period. So after the stimulus, just to see basically once we did the stimulus presentation, how the cortical activation pattern differs. And one thing that we found here was that there is a difference in the selectivity for which side is the correct side to choose, given the sensory information. Where in the visual trials, we actually see activation of these secondary visual areas, In this case, we focus on this area called RL, it's creatively named rostrolateral visual area. Rostrolateral basically just meaning in the back and to the side. And in that area, you can actually see that even after the end of the stimulus, there's more information about where the correct side would be. So this is also visible in these traces here, where you can see this blue trace kind of going up versus in the tactile case, it just goes down. So it seems like there's a signature of the information that was presented during the stimulus period that basically is still present during the delay and probably important for the animal to know what it should ultimately choose. When we look at the tactile trials, there's more information about the correct side in these frontal regions. So in this case, this is called the medial motor cortex, where we see that the site-specific activity is enhanced. Although it's not super clear because in that case, we don't get this clear peak that we saw for vision in the parietal cortex. But the key thing here is basically that they differ in terms of which regions seem to be most sensitive to behaviorally relevant information. And so I'm going to basically conservatively say that there are some modality-specific differences during the delay period, and it suggests that there could be different pathways for the accumulation of sensory information to guide behavior, right? So the information isn't immediately combined, and then there's a single integrated area that just goes up, but it seems like it actually remains in separate cortical areas for longer amount of time, like even after the stimulus is over. And so then we can again turn to choice signaling. So this is again the choice decoder, as I showed you earlier, still has this kind of ramping activity that increases over the time of the stimulus presentation. And we can look at the weights. And in that case, it's a bit less obvious in these data, but we do see the activation of this frontal region again as the stimulus is being presented. So this is shown here in blue. And in contrast, when we look at other regions, it's less clear, even though there are obviously other areas that also have choice-related information. And we can then compare these choice decoder maps across the different modalities, just to ask whether these are also modality-specific. In that case, the answer is no. So when we look for choice signals specifically, it seems like these decoder maps come up pretty similarly. So the way that we interpret this is that even though there is stimulus-specific integration of the information, it then turns into a unified choice output, right? So basically, on the one hand, we have specificity in the representation of sensory information, but it's unified representation of the animal's choice. And we can turn this into this little toy circuit for the cortex, where the idea is that visual information could be integrated in the parietal cortex, and tactile information could be integrated in the frontal regions, but then at some point, they're being unified into like a choice area. And then we can turn back and do optogenetics on this. And with the optogenetics, we can do inactivation of either this parietal area, where we then see a strong impact on the visual performance during the stimulus. Oh yeah, and there's a weaker effect when we turn to the delay. So this is in line with the idea that this is more sensory processing rather than choice. Or we can inactivate the frontal cortical regions. In that case, unfortunately, we didn't have enough resolution to separate between these two. So in that case, the fiber is actually on this region, a bit more in the front. But we get this clear tactile impact during the stimulus presentation. So during the stimulus presentation, there's an impairment that is more tactile than visual. But then when we move towards the delay, so closer to the choice, we have this more unified impact on behavior that kind of hits all the modalities pretty equally. So yeah, so we see this overall in line with what the image suggested is going on. So the last thing I wanted to mention is that aside from the imaging, we can also use electrophysiology. And the cool thing about the EFIS is that you can use these probes. So like this is called a neuropixels probe that are spanning most of the mouse brain. So you can see this is a very long shank, and you can then basically really record the activity for many different brain areas at the same time. And not only can you do this, you can also combine it with the cortical imaging. So you basically get both the cortical and the subcortical activity in conjunction. And so this is an example brain slice where we see the placement of this probe because we put a fluorescent dye on it. And then we can reconstruct in 3D where exactly these probes were localized in the brain. And I'm just going to mention that this data exists. I think this goes a bit far. But basically, when we do these electrophysiological recordings, we can also ask how individual neurons in the brain are responding to different parts of the trial. And we see that, for example, there's visual tuning in the primary visual cortex and parts of this midbrain area, but then we also see choice-related signals in deeper parts of the midbrain and this frontal cortex. And we can again do optogenetics, and everything is really great. This is basically just to say that we had some conclusions and we did some optogenetics, and they seem to be, again, pretty much in line with what the data suggest. And I will jump over this part and maybe end on this part saying that one thing that was interesting when we looked at the choice coding in the frontal cortex was that it seemed like in some areas, the neurons that are choice sensitive depend on the modality that made the choice. So it seems like in the frontal cortex, there might be neurons that are visual choice neurons, but not tactile choice neurons, which goes beyond with what we saw in the imaging, because there we didn't see a specific signature like that. So I mentioned that just to point out that when you increase your specificity and you start really recording individual neurons, you can see additional information that isn't visible if you just have a population readout, because all of these things otherwise get averaged together. Okay, so to summarize this part, so I showed you some of the photometry, so we can use that to study projection pathways, especially when it comes to cortical-subcortical interactions. We are interested in multisensory integration, especially when it comes to formation of multisensory choices and leads to these toy cortex circuit models. And we can use the EFIS to study the activity of these deeper brain regions more directly. Yeah, and with that, like, I mean, thanks all of you for your attention, and obviously my group and like all the people that do all the work, especially Anushka, who is with us today, and we'll talk about some of her work as well. Yeah, thanks.
Hello everyone, I'm Anushka. I'm a PhD student in Simon's lab. My presentation is not as beautiful as Simon's presentation is. Like, his presentations are usually very spectacular. So, so here, like as Simon said, that in our lab, we do a lot of electrophysiological experiments. So the one issue that is with when you're doing electrophysiological experiment is to find units which are on a single cell resolution, and those are actually single cell. I will tell you like why that can be an issue, but to do that, we created a toolbox called Unit Refine. It is like a community-based toolbox, where the aim is to automate spike sorted curation. So we are doing neuropixels, and the good thing about neuropixels is that, as Simon said, it can penetrate through the whole brain of the mouse. And the good thing is that we can record from a lot of neurons. Like 1 hour, we are usually doing recordings which are an hour long, and in that hour we have like 700 to 800 clusters. What I mean is that we think that those are neurons because it cannot, like, we think those are neurons, but we have to do a curation. So the idea is that we have this pipeline to do to look at the data. So the idea is first you have the raw signal, then you do some pre-processing, then you get a spike, then you do spike sorting, and the output of the spike sorting are neurons, which are like putative neurons. You have to do a lot of manual curation to see that if they're actually neurons or not, because sometimes they can be noise, or like sometimes they're false positive that a spike sorted thing, it's a neuron, but it is not, because it's a huge amount of data and spike sorters can make mistakes. So what you usually do is you go to a GUI when you're doing manual curation, and you look at their waveform, you look at their ISI, like the correlation values, and then you look at their amplitude over time. And then we saw that in our lab, we saw that the amount of time we are spending and the amount of units we are labeling for noise is huge. So then we thought that, is this like something specific to our lab, or other labs are also facing the same issue? And we saw that irrespective of you're doing neuropixels, like even if you're doing other kind of electrophysiology where your probe is not is not as highly dense, does not have that much of high density. Like NeuroPixels have 384 channels, but if you are using probes which have like 120 channels, you still have to do manual curation. It's still time consuming, and the results that you get are not reproducible, because like in some labs they have very strict ways of saying that this is our single unit, but in some labs they can be a bit lenient. Also depends upon the kind of experiments they're doing. So the one thing that we wanted to do was to have a supervised machine learning model to automate this manual curation. To do that, we labeled 11 recordings from our lab, and we put a lot of effort so that the people who are labeling, they have some agreement, because we want to see that if the labels are done well, and then we can get some features from the units that we're seeing on the GUI, can we create supervised machine learning algorithm for it? And this is what we saw, that like we had 2 to 5 people recording, labeling each recording, and we saw that they have good accuracy among the curators. And the cluster distribution in our data set, we saw that more like there is noise, there is also a good amount of SUA, and like a good amount is also like multi-unit activity, where it is not a single unit activity, but the neuron, like there are multiple neurons that are captured on a channel. Are there any questions? Just also interrupt me if you have any questions. So the one validation approach, we wanted to see that can these labels be used to do a supervised machine learning on the data set. So the first step we wanted to see was the information that we're getting, is that redundant or not? So what we did was that we looked at different features which Spike can give us, like peak to trough ratio, repolarization, slope. So these became our features. And we also had features which were like describing spike, like interspike interval. All these matrix we combined together, and we wanted to see is there information which is independent, and that is something that we were very happy to see, that all the metrics that we have do have independent information, and we can use these metrics now to have a machine learning algorithm. Another test we wanted to do was to plot a ROC AOC. So that just means that we wanted to see if we have a metric, can it be used to differentiate between two populations? The one was SUA and the other one was not SUA. So there we saw that that there are definitely metrics which are discriminative. So another question that comes is that which algorithm should be used to do supervised machine learning. So there we benchmarked a bunch of classifiers which exist in sklearn, and then we also benchmarked it with neural networks, transformer-based classification, and then bombcell is also a toolbox which is based on thresholding. So we saw that random forest is performing really well in classifying noise and also SUA. And we saw that the random forest is giving really good performance. And another thing we wanted to see was how much data do we need, because we don't want it to be that, okay, there is a classifier, it needs a lot of data, a lot of manual labeling. We wanted to have the algorithm which does not take too much of data and still can give good performance. And you can see that the random forest, like if you give 500 units, it's performing really well. So like we see that random forest outperforms other classifiers. And the one thing is also good with random forest that it's very easy to implement. Like you have the sklearn API to use random forest. So the next thing that we wanted to do was, okay, in the previous slide I showed you, we had the classification for noise versus not noise, and then it was SUA versus MOA. So one thing that we saw when we were talking to other labs is that they usually do binary decisions for all their data set. Like they're not doing it in a hierarchical way that we wanted to try. And even in a binary fashion, it works really well. And one thing that was also important to see was which metrics are important to do a good single unit classification. So we used SHAP. It is a toolbox where you can see which feature contributes most to making a decision. And we see that ISI violations, RP contaminations are important to make this decision. And it also is good that it also validates like what would you also imagine. So this was like a good result. And are there any questions? Okay.
Anushka, I have one question actually.
Yes.
Also, a bit prone to overfitting. How did you manage that one?
I cannot
Did anyone hear me? My question can maybe repeat.
What's the question? How do you deal with potential overfitting in the model?
Yes. So that's a great question. So what we do to account for overfitting is the results that we get are cross-validated. And the good point that I think I forgot to mention was we do the cross-validation is also in a leave one out recording. So what we're doing is if we have 7 recordings, we are training on 6 and predicting on 7. So the data points that you see are then on those 7 recordings. So in the whole process, what we usually try to do is if there is a new animal, if there is a new brain region, we tried in all those ablations to see that if the unit refine would perform well. And that is what we saw, that using a classifier, which when it's trying to find which model works best, there is also, we have an internal cross validation to figure out which data, like which machine learning algorithm is best. That is random forest. When random forest comes, then when we see that, then we want to see if it performs well on different brain region. We also saw if it's different probe, different animal. So we saw in all those cases and it worked really well. Yeah, and also that is why another thing that we wanted to see was does it generalize well on different data sets? So we got 8 recordings from IBL. And here what we did was, so the base data set is the data set that we have in our lab. And what we did was we trained on the base data set and predicted on the 8 IBL recordings. It worked really well. And then what we also saw that if we test on IBL data, which that means that we train on IBL dataset and predict on IBL dataset, then the performance shoots up. So what that means is if you're using unit refine, you can use the data from our lab and predict on your own data. If it's the same species, it works really well. But if you want to have like better performance, then it's probably good to also retrain it. Similarly, that is also we saw on, oh, I think I missed it. So this is also we saw on Allen dataset here as well, that with IBL, with Allen, and with our base dataset. So this we did it in a leave one out session. Again, what this means that we combine all 23 recordings and we trained on the 22 recordings and predicted on the 23rd. And all of these recordings were from different labs. One was from can be from IBL, Allen, and Base. And in all situations, it performs really well. In few of our labs, it was around 70, but for most of the recording, it's performed really well. And the good thing that is also we can see is that the metrics which we see are important for the classification are again consistent with what we thought, like ISI violations, RP contamination, half-width. These are the metrics which, even when you're using thresholding, you put usually threshold on these kind of metrics to isolate single unit activity. So that also goes in line, like when you're using some bombs, like bomb cell kind of approach, which is based on heuristics. The results that you get from unit refine and what you would get from using thresholding would align. And this another thing that we wanted to see, that if it generalizes across different brain regions. So here again we did a leave one out cross validation, and we saw that for Allen dataset, if we want to see that, okay, if we have dataset which is trained on all these 4 regions and we want to predict on cortex, it also performs really well. And these are the sharp rankings. It's similar to what I told you, and here we can see it in a format which is easy to understand, that for all of 3 labs, and if we combine all of the 3 together, it's similar that there are a few metrics which are really important, and these are important for all the data sets in the mouse model. And the one thing that we wanted to see, that we wanted to see if this performs also well if you're not using neuropixels, or if you're not using like in the mouse model, we're doing some experiments on mole rats or on monkeys or on humans. So does this approach generalize there as well? So for mouse, so for mole rat, we saw that if you use a pre-trained mice model, it performs well. And if you also then do if you use a model which is trained on mole rats, it also performed well. And the very interesting result was that if we combine mole rats and mice dataset, you also get good results. And this is similar to, like, in all the species, like if you combine with mice with monkey and mice with humans, you do get good performance. So like if you are a lab who are doing EFIS in different species and you have limited amount of training data, like if you have labeled like few recordings, so if you combine the data from our lab and then make a model out of it, it should perform well. Yes, so as I said, that retraining on species specific labels improves the accuracy. Yeah. And here we also see that, again, for all the species, ISI violation ratios is important, firing rate is important. And another cool result was that if we are combining these data sets together, and then we want to see that, okay, is there like a generalized model that exists, so that if we have data set from different probes, different species, and also like then it would be also different brain regions, then our poll data also performs well. That here also it's a leave one out session. Yeah, I think there was a question.
Actually, I have a question about
Yeah.
the right graph of the figure.
Yes.
Difference between the numbers and the color. There is a difference, right? So the color is the importance of the fact that it has that number, or
And yes. So the number of data points that I have is depending on the number of sessions I had for that species. So
I meant
for
the
human, I'm
ranking, the right one.
yeah.
This one.
Okay.
So you have these numbers, right? So
Yes.
one, I think the highest is I've seen is 12, right?
Yes.
What do they so is that how they rank among each other? And then how does the importance, because you have these numbers, right? And then you have a color scale as well. And I wanted
Yes.
to how these two relate to each other.
I think I'm having some issue in hearing or
But
understanding.
I think the general question is how these numbers are to be interpreted, right?
Yes.
I think if you explain that, the rest will be clear.
Okay. So the way I plotted this was that like I showed you in my previous slide that we have a sharp performance. I can maybe go on this slide.
Yes.
So here you are seeing that so these are cross-validated. The bar is like, I think it is for each recording, and what I did was you get this mean sharp value for each of the sessions, and then I rank like it's like the first rank is then giving to the amplitude median, the second is giving to the ISI violation, and then this ranking I put it in a tabular format.
Okay.
That makes sense. Yeah.
And so the numbers denote the ranks.
Yes, exactly. Those are the ranks. Yep.
Okay, and then the color scale that we have.
Yes. So the color scale that I have is from like the darker they are, the important that feature is for the classification. And so what I mean by that is for the mouse and MORAD, like ISI violation is that a very important metric to do a correct classification. And for like human, it is sync spike 4, and for monkey, it is RP violations. And what I can say that the firing rate is one has like it is like if you sum it, that has the minimum sum.
But I think that the short answer here is that the color and the numbers mean the same thing, right?
Yes.
Okay.
Yeah, I think so. The colors are just a visualization of what the numbers are telling you. So you can basically look for the lowest number for a given species to identify which feature is the most important because it's ranked first.
Yeah.
Okay, yeah. Makes sense. Thank you.
Yeah, and I also the point for this is that sometimes people are doing EFIS and then they're doing it in another species, and then it's also like from experimental standards, like they cannot compute all the quality metrics or they cannot compute everything. So this also shows that there are certain quality metrics that you should definitely have in your training model, like when you want to do a classification.
Okay. Thanks.
And yes, so like a unified approach is good and it also works. We were very happy with this results. And another thing which was a very important question was, okay, you get units out of after using unit refine, you get like some units which are supposed to be single unit activity, but then is there a real impact of using it, or it's like even if you do threshold it's similar? So this results tries to quantify that, the like if you have a mouse which is doing a task, and so it shows that I think this result is out of 699 IBL sessions. So like huge amount of recordings are there. And what we see is that using unit refine, you do get a lot of single neurons out. And then the idea is, okay, the mouse is then doing like a perceptual task, like a feedback or a choice. So does using unit refine help? Like that curation helps, or and it shows that if you're using unit refine, then like you get higher decoding accuracy in both the feedback and choice, and also like the subregions in like the accuracy in identifying those subregions, which are then a part of these scores are also important, and with unit refine that is also there, like you can find more subregions which are then predictive of the behavior. And another way of visualizing is through this whole brain maps, and the idea is that the color which are in red are more indicative of improvement in performance, and the blue ones are like the red ones are better performance. And here we see that the red performance, the that you're seeing is not on a few regions, it is on a couple of brain regions. So that just shows that if you're using like unit refine, that really helps like on a large scale in identifying brain regions. And it is something that we saw like works. And yes, so in the end, what we wanted to do was that we identified that the approach works well, and we wanted to have it in a way that how like it's easier for other labs to use it. So for that, we created a unit refined GUI. And the idea in that GUI is that it has this active learning framework. And the idea is that you would use you would use the GUI, and you would label a few sessions, and then you can train a model, and then you can push it to like Hugging Face. And then if you want to reuse that label on a different data set which that model has never seen before, then when you apply it, the GUI can identify certain units which have low prediction confidence, because we're using random forest. With random forest, you can get a label and also a confidence with that label, that how confident the unit is. And then you can manually label those units, and then you can keep on updating the your training pool. And the idea is that you can improve your classification training model iteratively over time. And we also think that if a lot of people would use it, and then they would put their data set on Hugging Face, then the idea would be that at some point we would have a training data set which is labeled by multiple people, by multiple labs, and it's huge. And then it should be like a unified model, and it should work well. So I think a lot of people have to use it for it to happen, but hopefully we reach there soon. So thank you for listening to me. And I would just say that all the models that I showed and like all the code is available on Hugging Face. We also have a paper for it. So if you have questions, if you want to try it, like you can read the paper and also the documentation. I have added a lot of tutorials, or you can also reach out to me. And if you're doing Mia recordings, which is not in the presentation yet, but I got a lot of feedback from people that they use Unit Refine on their Mia recording session, and it works really well for them. So if you are doing Mia recordings, just reach out. I can help with that.
Any questions?
Yeah, so I have one.
Yeah, please.
Did you compare different models or just random forest? So why did you choose random forest instead of other like gradient boosting or?
So this like I can show you this result. I can share my screen. So this is what we saw, that like I try to benchmark it, because this was the question that I also had. that, okay, why do we want to use random forest and why not any other classification? So we saw that random forest works really well, and what I saw that it also worked well for other species and other labs. But what you can do is, if you want to try it on your own data set, you don't have to stick with random forest. So in the API of the unit refine, you can give it a string of classifiers that you can say, I want to compare it between random forest, arabos, and like a logistic regression, let's say. And then it would give you an output on like which classified it thinks is the best for your data set. It would do that based on like a cross validation, and there also you can specify how many folds do you want. So the API is in a way that it can figure out which classifier works for your best data set with which hyperparameters, with the what scaling or with imputation. Like if you like, it can give you all those solutions. You don't have to rely on random forest. If there is other classifier that works best for you, you can definitely go for it. But I saw that it was random forest was like giving pretty good results for all the species, so that is why we're using it.
Thank you very much for your presentation.